And in doing so, let's take a step back from this word fertility and ask, what is fertility and fertilization really all about? Now, there are two general types of cells in the body. The egg in females and the sperm in males are part of what's called the germ line. It's just that the cells of the germ line have genes that cannot be modified by the behavior of the individual that houses those genes. But that's all occurring in the so-called somatic cells. Okay, so the key first thing to understand today is that there's a distinction between somatic cells, which is the vast majority of cells in your body, and the so-called germline cells, which are the egg and the sperm. So when each one of the eggs that a woman has and that she's had since she was an embryo, and that's contained in this vault, those eggs are, of course, going to be very immature at birth, right, called the diploid, and that means that they have 23 pairs of chromosomes, as distinguished from cells that are haploid, where there's only one set of those 23 chromosomes. So instead of 23 pairs, there's only 23 chromosomes. But right now we're just talking about the egg cells. And you have to get the egg and the sperm in proximity so that the egg can potentially be fertilized by the sperm, bringing the DNA, the 23 single strands of chromosomes from dad, into a cell that has 23 single strands from mom. So the ovulatory cycle and the menstrual cycle are really about first eliminating half of the chromosomal pairs in that 23 sets of chromosomes and not getting rid of, for instance, half, just going 1 to 11 or 12 to 23. That's not the goal. The goal is to have chromosomes 1, 2, 3, 4, 5, 6, all the way up to 23, but only to have half of the chromosomes there, and to bring that cell together with the sperm cell physically. But I'll just highlight a few things that we know about the onset of menses or menstruation, or it's sometimes also called menarche. It doesn't necessarily mean that something bad is happening. What do we know for sure? Well, we know that one of the things that can trigger the onset of puberty, in particular in females, is that when enough body fat accumulates, that body fat releases a hormone called leptin. And that hormone leptin travels in the bloodstream across the blood-brain barrier and goes to the hypothalamus and can trigger the onset of puberty by activating the neurons that release gonadotropin-releasing hormone. Now, I can already imagine a number of people are thinking, this must relate to the obesity crisis. And indeed, there is a crisis of obesity. I want to review some data that talk about the onset of menses, menstruation, that is puberty, in females according to country and according to age over the last 100 or more years. What we see is that the average age of the onset of female puberty in 1850 in Norway was 17 years old, whereas in 1970, it's 13 years old. So if we were to look in, for instance, in the UK, they have a smaller data set, meaning it only extends back to about 1940, but the average age of the onset of puberty in the UK in 1940 was 13 and a half years old. Unfortunately, they didn't continue to collect data out to 2022. But I'd be remiss if I didn't tell you about a certain result which shows that if a young female is exposed to the odor, not necessarily the pheromones, right? And we think also that is not biologically related to her, can trigger earlier onset of puberty. And that effect can at least be partially buffered by her being in the presence of the scent from her biological father. But those two hormones are called luteinizing hormone, which is abbreviated LH, and follicle stimulating hormone, which is abbreviated FSH. Okay, so the simple picture that you need to have in your mind is gonadotropin releasing hormone from the brain, from the hypothalamus in particular, is causing the release of lutein. Okay, so we're now going to talk about ovulation and menstruation. We would just call it the female reproductive cycle. There are some females for which the ovulatory cycle will be shorter. It can be as short as 21 days. One of the key things when thinking about fertility is if you talk to OBGYNs who are focused on fertility, which I have in anticipation of this episode, they'll tell you that whether or not your cycle is 21 days long or 35 days long is not as much of an issue necessarily unless it's happening to become much shorter or much longer in a kind of erratic way. But this is pretty well established as far as I know. Okay, so we need to bring together the so-called haploid contents, the 23 individual strands of chromosomes from the egg, to a place in a position where it could potentially be fertilized by the male. There's a lot of blood supply to the ovary. And FSH and LH arrive at the ovary because of this relationship between FSH triggering the maturation of a subset of follicles. Now, we'll get back to that egg in a few moments, but the way I'd like you to think about the pituitary for sake of feedback loops is that it's sort of like a thermometer that you would put into a pool, like a backyard pool, that is attached to the heater. The pituitary says, okay, and releases more follicle-stimulating hormone and luteinizing hormone, and bam. The second half of the ovulatory menstrual cycle is the luteal phase because of corpus luteum. I think if you can just generally understand that the first half of the menstrual ovulatory cycle is marked by the maturation of the follicles in FSH, and that the second half is marked by the accumulation of progesterone and the thickening of the uterine lining should fertilization and implantation occur. One of the, I view it as an advantage, but one of the things that many females can really do and experience because they have cycles that occur every month that are fairly dramatic in terms of the levels of hormone. So for instance, a more than 1,000-fold increase in progesterone during the luteal phase of the menstrual cycle. It is a signal from the brain, from the hypothalamus, which then triggers a signal from the pituitary, an endocrine gland, which then signals the release of hormones that travel to the ovary and that control two things: maturation of eggs and the identification of one egg in particular, and then preparation of the milieu, the environment in which that fertilized egg could potentially land and mature into a healthy embryo and child. So if you have that framed up in your mind, and even if you just extracted maybe 10 to 15% of the hormones and different aspects I described up until now, I would consider you far more knowledgeable about this entire process than 99% of people out there. Certainly not the OBGYNs and urologists, but than 99% of individuals out there. Talking about the generation of sperm cells, what we're really talking about is FSH for stimulating the maturation of the sperm cell. There are instances where the testes fail to descend during development. We'll talk about that a little bit later. Well, then oftentimes the sperm that develop are not healthy. Not healthy in a number of ways. But just to give you a simple takeaway, if you are hoping to conceive in the next 90 days, and it turns out the cremaster muscle can achieve that whether or not people are wearing boxers or briefs, although it stands to reason that any kind of, there's no other name for it, undergarments, I don't know why that word is it just seems kind of antiquated. You have the cells, the so-called spermatogonia, which differentiate into so-called spermatocytes. You don't have to remember all this. And the spermatocytes undergo this process of meiosis. Meiosis is a form of cell division, which reduces the chromosome number to those 23 individual strands as opposed to pairs. Well, it turns out that the function of the spindle in the egg and the sperm is heavily dependent on mitochondrial function. But when you talk about it, you talk about if you were to take a visual cross-section through the testicle, what you would find is that there are a lot of different little tubes, a lot of ducts. Now, this will also become a relevant conversation, and they always check to see if zero sperm and confirm that zero sperm are being released in the ejaculate. There's some general rules that, of course, come to mind, which is ejaculations close to ovulation, both before, during, or sometimes after, are obviously advantageous. So if you're a male or if you're a female, men can generate sperm their entire lifetime. Okay, prior to that, testicles are present, but they're not generating sperm. Ejaculation isn't possible, or if it is possible, it's very unlikely and unusual. Okay, all the things we associate with puberty and with testosterone typically. Restricting the conversation to the effects of testosterone on the testicle itself and on spermatogenesis, the Leydig cells make testosterone, and keep in mind that some of that testosterone will travel elsewhere in the body and do its thing for gene expression and the more acute effects of testosterone on the brain included. Cells, which make androgen binding protein, testosterone and androgen binding protein combine and create a chemical and actually a structural milieu in which those little spermatocytes can go from the walls, from literally the walls of the tubes of those seminiferous tubules, can mature into healthy, well-developed sperm and can hop into those ducts, those little tubes, and then head off to the epididymis where they will reside. 2 is somewhere in between 1 and 3. One thing I didn't mention before when discussing the production of eggs and ovulation, and I probably should have, and indeed sperm counts do seem to be declining. Okay, so the idea here is that we want the maximum number of high quality sperm, that is rapidly forward motile sperm that are of the correct morphology, that is shape, That's going to require a lot of mitochondria in the mid-region, a well-shaped head. But of course, there's the female side of it too, which is that ovulation occurs on just one day during the menstrual ovulatory cycle. And this is where it's really useful to have a cycle. Here, I'm only referring to intercourse as it relates to the goal of fertilization of the egg. Okay. So knowing what we know about spermatogenesis and the fact that ejaculate is going to contain a certain concentration of sperm, but that within that ejaculate, some of the sperm will be older and less healthy, and some will be optimally mature, and some might even be a little bit immature, although there's a tendency for the immature sperm to not have yet exited the seminiferous tubules, gone into the epididymis and vas deferens. And given that with each successive ejaculation in a short period of time, there's going to be a decrease in the concentration of sperm per milliliter of semen, of ejaculate, we can make some good arguments as to how often couples should have intercourse with ejaculation around the time of ovulation if the goal is to fertilize. If ovulation occurs on, for instance, day 14 of a cycle, but for some people with a 30-day cycle, that could be day 15, or with a shorter cycle, it could be day 12, for instance. So what this involves generally, and what the typical recommendation is to abstain from intercourse with ejaculation about 2 or 3 days out from ovulation. And then on the day prior to ovulation and on the day of ovulation to essentially introduce as much semen and ejaculate into the reproductive pathway of the female as possible. What that means is that if a couple, for instance, were to have intercourse with ejaculation many times on the day prior to ovulation, you'll see a lot of information about this online, but there's a lot of misunderstanding about it as well. The female, in particular, a change in the pH, that is the relative acidity versus basic nature of the mucosal lining near the cervix, and also vaginal secretions, such that around the time of ovulation, the entire milieu of the vagina and the cervix, while it's not the only factor, is certainly one of the most determining factors in whether or not successful fertilization occurs. And as women age, the quality of the eggs tends to diminish over time, largely due to changes in the mitochondrial function and the spindle that pulls the chromosomes apart, although there could be other factors involved as well. So when we talk about egg quality or sperm quality, we're really referring to an amalgam of different features related to the different aspects of ovulation leading to successful fertilization, or the different aspects of sperm related to whether or not they can arrive and deliver their DNA contents and so forth. Now, if fertilization and pregnancy occurs, great, right? There'll be at least a 9-month lag until they decide whether or not they want to try and conceive again. Great. And if they don't, then they ought to try again the next month, and so forth. However, when it comes to fecundability, we're really talking about something which is called cumulative pregnancy rate, which is not really independent probabilities. It is, in principle, receptive to fertilization, so that could limit the ability for that to happen. But in general, that's why OBGYNs will tell their female patients, look, if you're setting out to conceive, so when the age of the woman starts extending out to about 34 or 35 years old, then the typical advice of the OBGYN is going to be to attempt to conceive over a period of about 9 months to a year before deciding to take some sort of medical intervention. There are many women who in their late 30s and 40s and even early 50s have successfully conceived healthy children. This is always an egg-sperm dynamic. And when I say egg-sperm dynamic, now hopefully that calls to mind the huge library of information that we've been covering up until now about chromosomal segregation and the coming together of these different cell types and their genetic information. Any number of different steps within the process of fertilization leading up to pregnancy can lead to miscarriage, such that by the time women are in their early 40s, so 40 or older, the probability of miscarriage after a successful fertilization is going to be about 50%. But one common reason is that there can be chromosomal abnormalities, and that could be related typically to the segregation of the egg when half of the chromosomes in that egg are removed, taking it from diploid to haploid. If you recall, there's that little polar body, which is the removal of the chromosomes that's ejected from the egg that will eventually ovulate. There are some instances in which all of the chromosomes and all the chromosomal arrangements are perfectly normal and miscarriages can still occur. One thing that you already learned is that there's this vault, there's this reserve that we call the ovarian reserve, and that each month a certain number of follicles leave that reserve, and there's the opportunity based on the ovulation of a single egg to fertilize that egg and for the woman to get pregnant. And typically this is done over a series of months to determine how many eggs are leaving the ovarian reserve each month, and therefore how many eggs she is likely to still have in the ovarian reserve. Now, the mirror image of all this, of course, is the sperm. And, there's a kind of common misconception out there that, the sperm, you only need one, and indeed you only need one, but actually you need many, and it's only one that's able to successfully fertilize the egg. But having that in reserve is generally a good idea. And I discussed some of the parameters that are looked for. As I mentioned earlier, the seminal fluid in the ejaculate could have 0 sperm in it, but the volume of ejaculate could appear completely normal. So just because your ejaculate volume is normal to you or is in that range of 2 to 5 milliliters, well, then that doesn't necessarily mean that there are any sperm there or that there are very few sperm there. And the few sperm that are there or the many sperm that are there are functionally motile. But the shape of the head of the sperm isn't quite right. And of course, typically with a sperm analysis and a ovarian reserve analysis will be a hormone analysis. And I'm a very strong believer in people getting an insight that is a window into their hormonal composition, even if it's only done once every 5 years or so, having a reference point to your 20s and to your 30s and mid 30s for when you felt a certain way. Maybe, as in the case for many people I know, they actually feel better in their 40s than they did in their 20s because they're doing a lot of things to support their health. That is possible. Now, there are other things to not do. And those, again, fall into the somewhat obvious categories. But I think a lot of people aren't aware of just how striking an effect these certain behaviors that you want to avoid can have in diminishing your fertility for both males and females. Well, it turns out that smoking increases what are called reactive oxygen species, an attempt to fertilize the egg and lead to a healthy pregnancy. Now, I am not somebody who believes that cannabis across the board is not useful. Well, then you should definitely be aware of the data showing that even just one bout of so-called binge drinking, which is 5 to 6 drinks in a given night, or half day in a 12-hour period, one bout of 5 to 6 drinks, if you're a woman or you're a man, greatly increases both the likelihood of mutations in the embryo that would result from a fertilization, and at the same time, for reasons that should be obvious to you based on all the biology we've talked about, a greatly reduced probability of fertilization. Now, that absolutely does not mean that you should use the ingestion of 5 or 6 drinks as a method of birth control. So those are the major don'ts. Really avoid excessive stress. And I should mention, excessive stress is not just best avoided by getting enough quality sleep at night, although that is the primary way. There are other ways to avoid stress. Again, you can find all that at hubermanlab.com. So reduce your stress. And as I just told you, by all means, do not drink more than 2 drinks per week, and 0 is better than 2. If anyone tells you, there's, all this resveratrol in red wine and that's good for us, the data simply tell us that there's not enough resveratrol in red wine to really have any positive health benefit. Not necessarily, but you should talk to your OBGYN about that. And so what that means is that these men can have what appears to be normal semen volume, but that they won't have normal numbers of sperm. And that's not because of a deficit in making the sperm. As I mentioned before, avoid going in hot tubs during the period in which you're trying to conceive children. The other thing that you'll definitely want to do is avoid putting a laptop or any other hot device directly onto your lap. But be aware that if you're somebody who's trying to conceive, you want to do whatever you can to reduce the temperature of the scrotum, or at least not let it get too hot for too long. Keep in mind, there is basically no controversy that radio frequency waves and EMFs can have a negative impact on biological tissues. And the question is, should you carry it in your front pocket if you're a male? I think to be on the safe side, the answer is probably avoid doing that too much of the time. Ideally, don't do it at all. Sperm quality declines when people start using a mobile phone. So from the point they start using a mobile phone, regardless of the usage time, this is important. So both of those factors, independently and together, of course, can disrupt the Leydig cells of the testes and the production of testosterone. And intratesticular testosterone is important for sperm production. So any discussion about heat and sperm and how heat is detrimental to sperm has to raise this issue of whether or not cold is good for the testicle. And people will say, well, how cold? I should have mentioned that before. How cold? there is no way I can tell you exactly how cold the water should be, because for some people, 60 degrees Fahrenheit will be exceedingly cold. For other people, well, it turns out when you get into the cold, you get a big surge in adrenaline and then dopamine, which is very long lasting, provided that's done in the early part of the day. So I would say, not too close to sleep. Then what you do is you restrict your maximum cortisol release to a period earlier in the day that reduces, that is, the likelihood that you would have excessive amounts of cortisol later in the day, which not only can disrupt sleep, but is correlated with a number of other hormonal effects that are not good for us and therefore not good for fertility. So for men, we talked about increased testosterone, improved sperm quality. Likewise, with females, deliberate cold exposure can be good for the overall fertility process, not because cold is good for the ovary or being cold is good for the ovary, or for luteinizing hormone or for follicle stimulating hormone or anything else like that. The next things that we'll talk about in terms of positive things or things that we can do in order to maximize fertility for both females and males are the things that you also generally hear about elsewhere. Right along with sleep and avoiding alcohol and avoiding nicotine and avoiding cannabis, avoiding excessive heat for the testicle, avoiding excessive stress. Well, exercise, and that is both a combination of resistance training and cardiovascular exercise, is going to improve the health of the mitochondria, in particular cardiovascular exercise. One thing that I know a lot of people are interested in nowadays is so-called intermittent fasting or time-restricted feeding. Time-restricted feeding can have certain positive outcomes for various aspects of organ, cellular, and tissue health. Okay, this is somewhat controversial, but there is growing evidence that by restricting your feeding window to say 8 hours or 10 hours or 12 hours, that it is better than if you were to eat over a longer period of each 24-hour cycle. Okay, so please, 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 if you're pregnant, do not jump on a time-restricted feeding. The simple thing to take away from this is if you're a male who's using, because you like it, intermittent fasting, so-called time-restricted feeding, and you're following an 8-hour or maybe even a 1 meal per day type approach, and you are ingesting enough calories to maintain your weight if you're already lean, or you are ingesting fewer calories than you are burning in order to lose weight because you are already overweight and you want to lose body fat, you're probably optimizing for all the things that you need to do in order to improve sperm quality and testosterone levels. You need testosterone production from the Leydig cells of the testes, and you need spermatogenesis to be supported by that androgen binding protein coming from the support cells, from the Sertoli cells. The most typical TRT approach nowadays is testosterone cypionate, which is biologically identical to the kind of testosterone you would make. Well, because of negative feedback loops, which you also learned about earlier, the testicles themselves shut down their own testosterone production. There are things that they can do to offset that, like taking HCG, human chorionic gonadotropin, which is just kind of a mimic for luteinizing hormone, to stimulate the testes to continue to make testosterone. I've talked about in other podcasts, such as Tonga Ali. It turns out that there are a lot of men and women taking Tonga Ali to reduce sex hormone binding globulin levels, to increase testosterone and estrogen in some cases, libido, and so forth. Those approaches, meaning supplement-based approaches to increase testosterone or free testosterone or some related hormones, are not going to shut down your own endogenous testosterone production and reduce the number of sperm that you make, or at least as far as we know, disrupt ovulation in any kind of way, provided that the dosages are within normal ranges. But again, the major point here is that for both females and males, taking supplements to support healthy hormone production, including things like Tonga Ali, is not the same as taking hormones or bioidentical hormones, which indeed can shut down your own endogenous production of hormones and thereby reduce both egg quality and the chance of fertilization and healthy pregnancy, and sperm quality and the chance of fertilization and healthy pregnancy. Anytime. Now, of course, there are instances out there of people that have XXY chromosomes or XYY chromosomes. So for instance, Aristotle himself proposed that if a man is thinking about himself and his own pleasure more than his partner and her pleasure at the point of ejaculation, then the offspring will be male. Aristotle also asserted that if a man is thinking more about his partner and her pleasure at the point in which he ejaculates, well, then the offspring would be female. Of course, we have zero reason to believe that there's any truth to Aristotle's theory. Now, again, there are zero data to support this. Now, this, of course, is done in the context of in vitro fertilization. What that causes is the maturation of not just one egg that would be ovulated, but multiple follicles and eggs, and then ovulation itself is suppressed also through the administration of exogenous hormones. The arguments that point to a potential mechanism are that, if you recall what we were talking about when we talked about sperm quality involves a bunch of different measures like concentration of sperm per milliliter semen, morphology of those sperm, how many are forward motile. I did not say that. What I said is that if you use cannabis once, then the sperm that are generated in the 60 days after that cannabis use are going to have a higher incidence of disrupted motility and perhaps morphology as well. So acupuncture can operate at the chemical level, impacting hormones. It can act at the mechanical level, impacting the different tissues through which the egg has to pass, and so on and so forth. Likewise, on the male side, acupuncture has been shown to improve semen volume, quality of sperm motility, etc., and in large part through changes in the neural pathways that innervate the very tissues and vascular input to the scrotum and testicles, because as we learned earlier, temperature regulation of the scrotum and testicles is so vital for getting healthy sperm and increasing sperm quality. In addition, there are good data to support the idea that acupuncture can increase levels of testosterone, free testosterone, and the sorts of hormones that are going to support healthy hormone production and sperm production in males. The first on the list of things that people can take in order to improve egg quality or sperm quality is L-carnitine. L-carnitine is present in various foods, in particular in red meats. It can be taken with or without food. It does not seem to matter. And when taken for a period of 30 to 60 days, does seem to significantly improve all the parameters that have been discussed for egg quality and sperm quality. There's no reason to not continue to try for pregnancy before taking L-carnitine, but L-carnitine is going to improve egg and sperm quality. And so you might actually take the stance that even if you don't have any problem getting pregnant, wouldn't you want to maximize the quality of the egg that gets fertilized and the quality of the sperm that fertilizes that egg? One important note, if you are going to take L-carnitine in oral form, in capsule form, it can increase something called TMAO. TMAO can cause stiffening of the arteries. You don't want TMAO levels to go too high. One way to offset the increases in TMAO caused by oral L-carnitine is to take 600 milligrams of garlic per day. I suppose you could eat cloves of garlic. The next item on the list of compounds that have been shown to improve egg quality and sperm quality, and quite robustly, and urologists that are trying to assist their male patients with fertility will suggest CoQ10 because, again, it supports the health of mitochondria. Mitochondria are so vital to so many aspects of the formation and fertilization of the egg and sperm. For instance, I talked about inositol, and in particular, taking 900 milligrams of myo-inositol prior to sleep, which is something that I do along with the other supplements that I take and recommend for sleep, such as magnesium threonate, apigenin. Now, one point of, I wouldn't say caution, but of note is that myo-inositol can reduce anxiety, and it can be a slight sedative, which is why some folks, including myself, take almost a gram, 900 milligrams prior to sleep in terms of the level of anti-anxiety and drowsiness that it produces. 1 to 5 grams per day of myo-inositol is what's suggested for both men and women wishing to improve egg health and sperm health. That is the threshold of the EPA essential fatty acids. It'll say, 1,500 milligrams of omega-3s, but that's not 1,500 milligrams of the EPA form. But 400 milligrams a day of tonga ali has been shown to increase free testosterone by way of reducing something called sex hormone binding globulin. It's also been shown to increase luteinizing hormone, the net effect of which has been described as a subtle but significant increase in libido and some of the other parameters associated with increased androgens like free testosterone in males and females. And like tonga ali, even if they're not seeking to conceive, there's no reason to think that tonga ali directly improves egg or sperm quality except in males. And so it's something that both men and women wishing to conceive and or optimize their fertility just as a general health parameter might want to explore. The other supplement that's been shown to improve both egg quality and sperm quality, but there's some really good research studies exploring the supplementation with Shilajit at about 250 milligrams twice per day. Now, one note of caution, if you're a woman and you're considering taking Shilajit in order to increase testosterone and follicle stimulating hormone, keep in mind that the ovulatory cycle is this very tightly regulated cycle in which you want, so this is pretty esoteric stuff when it really comes down to it. So zinc is correlated with increases in breeding, but you never know which direction that correlation is running. Turns out that zinc in both animals, including fish, other mammals, and in humans, strongly impacts the enzymatic functions in the testes, including the function of androgen binding protein. And those other things generally are prescription drugs, rescue spermatogenesis, although not to the same degree as if people are not taking exogenous sources of testosterone. Some men, even if they've never touched TRT or exogenous testosterone of any kind, will be prescribed to take HCG because of its ability to stimulate the testes to produce more testosterone and sperm. And some are, for instance, outpatient procedures. And again, if you're somebody who's interested in conceiving, or if you've already conceived children, and even if you don't want more children, this is really the aspect of our biology that allowed us to be here. It's the aspect of our biology that determines so much. So hopefully that information was in and of itself interesting and perhaps useful as well. If you have questions or comments about the podcast or guests that you'd like me to bring on the Huberman Lab podcast, please put those questions and suggestions in the comment section on YouTube.